Our topic today is enhancing reproducibility through rigor and transparency, including sex as a biological variable. And our speaker today is Dr. Tonette Krauselwood. Dr. Krauselwood has many roles at Tulane, including Professor of Medicine in the School of Medicine, Professor of Epidemiology in the School of Public Health and Tropical Medicine, Associate Provost for the Health Sciences, Senior Associate Dean for Faculty Affairs, and Associate Dean for Public Health and Medical Education. She also leads the MD-MPH program. Dr. Krauselwood holds an MSPH from Tulane and an MD from LSU. She is and has been a PI and co-investigator on many federally funded grants, and notably, she is currently the PI and research director of the NIH's Building Interdisciplinary Research Careers in Women's Health Career Development Grant. Her research interests are focused on clinical outcomes and healthcare processes for patients with chronic diseases, such as hypertension and diabetes. Dr. Krauselwood has more than 135 publications, including peer-reviewed articles, reports, and book chapters. She is the president-elect of the American College of Preventative Medicine. She is a fellow of the American College of Preventative Medicine and the American Heart Association. Her national leadership positions have included standing member and chair of the NIH Study Section Health Services Organization and Delivery, member of the NIH Center for Scientific Review Advisory Council, chair of the American Board of Preventive Medicine, and a member of the NIH Center for Scientific Review Advisory Council. We're really delighted that she has made time in her very busy schedule to speak with us today, and I ask that you all please welcome her.
Well, thank you very much, Lisa, for a very kind introduction. Again, I apologize for some of the delay, and hopefully, as they would say in the airlines, I hope we can make up some of the time in the air. But today's focus is to build on what you've already heard on enhancing reproducibility through rigor and transparency by focusing on sex as a biological variable. So we're going to cover a number of things. We're going to talk a little bit about the background that established this SABV policy at NIH. Talk a little bit about some of the evidence about sex and gender differences in disease and how it presents and how individuals respond to it. We'll talk about some of the NIH perspectives and guidelines, approaches to SABV, as well as some of the resources that are available to you through the NIH and beyond. So this brief introduction will cover from the inclusion to our current status of a requirement of sex as a biological variable. And in the intermediary is where the science that supports the evidence for sex and gender as a biological variable has emerged. So this whole process of focusing on women, and I must admit it also includes minorities, was the NIH Revitalization Act in 1993. As prior to this time, the majority of the clinical trials that had been conducted in the US were focused on primarily middle-aged white men. However, once the evidence was generated from various trials, the therapies, whether they were pharmaceuticals or diagnostics or intervention procedures, these were applied across the board. So although they were done on middle-aged white men, they were used in women, minorities, and even children. So as a result of this act, it required that women and minorities must be included in all NIH-funded clinical research studies, that the trial must be designed to permit a valid analysis of the effects in women as well as in men, and that individual scientists who were submitting their NIH applications could not say that cost was an acceptable reason for excluding women or minorities. So therefore, we moved forward with this inclusion. Just to show you some of the recent data that has shown a result or an effect of that Revitalization Act in 1993. You can see the most recent reported was fiscal of 2016, and you can see for all studies across the board, we're actually seeing a higher proportion of women reported in NIH clinical studies. But if you look at those that after they exclude male only or female only studies, you can see we're right at about the 50-50 mark. over recent years and including the most recent one. So this is what we would expect, given that about 50% of our population includes women. So sex and gender are two terms that oftentimes we use interchangeably, yet they are quite distinct. When you look at the term sex, this is a classification that is derived from the chromosomal complement that each and every individual has. This is in essence a biological construct. When we talk about gender, and you'll see here it's represented by the symbols above it, and reflects more of a person's self-representation as male or female. This is more of a cultural concept that we adhere to. So when we look at how sex and gender influence health and disease, it's important to consider the things that are relevant to both but are quite distinct. And when you look at sex as a biological classification, it's important to remember that every cell in the human body, in your body, has a sex. Every tissue, every organ, because every cell has a sex. And that's important to know, and it's dependent upon whether or not you were genetically outlined as a man or a woman. Gender, however, refers to the socially constructed roles, behaviors, and expressions. This identifies girls, women, boys, men, and diverse gender people. It influences how people perceive themselves and how they perceive others, and how they act and interact with one another. So there's considerable diversity in gender, whereas for sex as a biological classification, it's one or the other, but gender allows a diverse understanding and exploration of how this might affect both physiological as well as cultural implications. So sex and gender, as we just outlined, has a profound influence on our daily lives. It includes not only the physiology, but also susceptibility disease. And as we'll talk about in a little bit, it also may have an impact on how individuals respond to disease. Historically, scientists considered that basic biophysiological processes were the same in females and males. But more recent evidence and research has indicated that that is not necessarily the case. So when we look at sex-biased genes, the question often comes up, where does it reside? Does it reside in the genome? Well, it's clear that although all sex differences stem from the X and Y genes, it's the sex hormones that cause sexual bias in the gene expression. And this acts directly on the genes through the genome. So when you're looking at the genome or the chromosome, et cetera, you almost want to ask the question, is it a chromosome or is it a hormone effect? Well, it's quite interesting that when one is looked after a gonadectomy, that almost all the sex differences in the liver gene expressions were abolished. So that indicated that perhaps the most important factors are caused by ovarian versus testicular hormones. So there are modest effects of the XX versus the XY genes or genomes, but what really happens is as a result of the influence of the sex hormones. So sex and gender research really goes beyond reproductive health, and that's a very important concept for us all to be thinking about, whether we conduct basic, translational, or clinical research and beyond. Certainly, the significant sex factors in diseased organs are related to reproduction, but there are others that are unrelated to reproduction. These issues or concepts beg the question, what are we missing by not including sex and gender in investigation at all levels? And what harm are we doing by not including sex and gender in investigation at all levels? So when we look at this, and we won't go through this in full detail, but just as an example, in looking at how sex and gender influence health and disease, you can see these examples. Sex would be indicated by looking at the chromosome markings, XX and XY, and gender is by the symbols of the male and female here. And you can see that across disease conditions, whether it's mental health, smoking or behavioral health, cardiovascular risk, osteoporosis, or knee arthritis, that there are differences both in the sex as well as the gender influences. We can talk briefly about the one for knee arthritis, that women and girls have a different biology with respect to the way the joint, the knee joint, is constructed, the way the tendons attach, the stability and the muscles around those joints. The differences in biology between males and females results in more likely injury to the knee in women and in girls than in men or boys. Now that's the sex difference. When we look at the gender difference, you can see that women are more likely, or those who characterize themselves as women, are more likely to wear high-heeled shoes. High-heeled shoes also have an effect on increasing injury to the knee, and that can have additional effect on gender expression, if you will, when looking at knee arthritis as it relates to cultural concepts. So there are other examples of sex differences and gender differences in other diseases as well. If you look, for instance, at arrhythmias, certain drugs like Seldane and Propulsid can trigger potentially fatal heart arrhythmias more often in women than they do in men. We know that women are more likely than men to have complications from implantable cardiac defibrillators. It's also have been established that women are more likely than men to experience surgical complications and more likely to have atrial flutter after they have an ablative surgery for arrhythmias. Important information for patients as well as their healthcare providers to have. We also know that risks differ in cardiovascular disease between women and men. We know that women, their cholesterol plaques are more evenly spread out the arterial wall, whereas in men, they tend to aggregate in sections of the wall. In women, aspirin reduces the risk of ischemic stroke, but in men, the low-dose aspirin has a positive effect on reducing the risk of heart attack. When we look at cardiovascular disease in women, the types of lesions that are more amenable to angioplasty are those that have segmental or single segmental stenoses, as indicated here on the left or in the diagram A. This is a more common presentation in men, whereas for women, the disease is typically more diffuse or have multiple stenosis, and this would not be appropriate for angioplasty. In the brain, we're also evidence also points to structural and functional dimorphism, both in human and non-primates, where we're seeing the influences of aging on gene expression are different in male and female brains. And in the work that leads up to interventions that can be used in humans, many of the studies are done in rodents. And already evidence is indicating that rapidly increasing reports of sex differences in the gene expression. Brain cells of female and male mice do not die the same way after a stroke. And this is giving us some of the initial, the early evidence that there may be molecular level sex differences that need to be explored and need to be considered. So based on a lot of this background information, in 2014, NIH came out with an article in Nature that kind of rocked the research world with the focus on balancing sex in cell and animal studies. So whereas I mentioned earlier in 1993, the NIH Revitalization Act targeted inclusion of women in clinical studies, it did not address inclusion or sex specification in basic science or preclinical studies. So here, both Drs. Janine Clayton and Dr. Frances Collin came forward with an article in Nature talking about the upcoming requirement that we include both male and female animals in these preclinical studies because ultimately the evidence that comes from them informs clinical studies and ultimately clinical practice. They clearly called out the fact that the basic science studies had an over-reliance on male animals and that they had neglected the attention to the sex of the animals and the sex of cells, as this might have important implications for research. So the evidence also showed in this article that there was underreporting of sex in animal studies, despite the fact that evidence was emerging that there were differences by sex in the progression of disease and in the incidence of disease. So you can see here in this diagram, if you look at this proportion of papers, there was a test mining analysis that was conducted and looking at data prior to 2014, showing that only 53% of the experiments using mice recorded both the sex and the age of the animals. And that's represented here in this dark blue line, where you have both sex and age reported. And you can see a much lower percentage of papers showing the reporting of only sex or only age. So it was clear that the male still dominated the animal studies. And I know in some of my early work, although I've been focused on clinical research the majority of my professional career, I remember my colleagues in the basic science, talking in the earlier days, talking about the fact that focusing on male animals only, made their studies more straightforward and simpler to complete, and not, and that was one of the reasons that that process was utilized. So here we can see in neurosciences, pharmacology, and physiology that the number of males to females truly outnumbers, with neuroscience being 5.5 to 1, pharmacology 5 to 1, and physiology 3.7 to 1. In surgical research, there was another evaluation of publications looking at the range from 2011 to 2012, assessing over 618 articles with animals and cells. And again, they found evidence that female prevalent diseases, that even in those types of studies, that sex, only 12% studied female animals, even for those female prevalent diseases, which of course leaves a lot on the table with respect to the evidence. So NIH came forward then, after this article that came out in Nature, giving people actionable items to strengthen the science in studying sex differences. And they outlined these 4 strategies that: 1, you have to first look at the problem; 2nd, identify a solution; 3rd, take action; and 4th, look at the outcomes. So in talking about the problem, it's important that we begin looking at female and male differences to avoid having a blind spot in our research and leaving gaps in research knowledge. The solution is that sex is a fundamental biological variable, must be considered throughout the biomedical research continuum, and that's in preclinical through clinical and population health studies. So the action that NIH took was to require a deliberate approach to consider sex in cells and animals to make sure that both men and women get the full benefit of all the medical research. And the outcome being that once this is considered, that this will help us deliver rigorous science and drive medical advances that benefit all people. So the NIH guide notices were released in 2015, outlining several new areas to ensure the rigor and transparency, and including the consideration of sex as a biological variable. And you can see in this as stacked bars here, it's represented in the 3rd one. The influence of sex and gender and going beyond just inclusion in clinical studies, you can see is represented here from the preclinical studies outlined here, looking at toxicology, the range of clinical trials, looking at sex-specific data analysis, understanding and educating about the impact, and then where we find gaps can go back to cell and animal studies, and the cycle continues. When we look at the phases of research, the first phase, of course, being discovery, including the basic or cellular and subcellular research, and including the basic research looking at cells, tissues, and animals. In both of these, the requirement is to observe, report sex-based data. When we go into turning discovery into improved health, there's the preclinical research, again, in the animal models, where we need to consider and include sex as a biological variable where appropriate. The translational research, again, to study both sexes as we apply what we've learned in the preclinical into clinical. And then in the clinical research, to test this in humans and where appropriate to include and study both sexes and examine the differences if they exist. And then of course in healthcare, as we apply it to humans, that we're able to deliver sex and gender relevant treatments. And then finally at the population level, looking at the treatment context, that we were able to display sex and gender informed care, practices, and policies. So as we go across this continuum, we're going from less data to more data and ultimately to improved health across the spectrum. So if you look at the impact on experimental design, traditionally, we looked at men and women aggregated and we compared treatments to controls, or males and females aggregated, and we looked at treatments to controls. If you look at this very simple diagram, you would see that the treatment had a higher disease impact than the control. And if that's a positive impact, you may say, wow, well, the treatment did something good overall. If you look at a more specific example, disaggregated by sex, a different message may reveal itself. Here in the example of looking at the effect of a inhibitor, PJ34, using male and female mice, and looking at the total who would have an infarct, right, to see whether or not this particular intervention would have an effect on reducing the total infarct. You can see here, if you look at males, there appears to be a dramatic reduction in the percent with a total infarct after using the PJ34. Whereas in female mice, it had, there was a dramatic increase in the number of, with a total infarct for those who were exposed to the PJ34. So combining them might have given us an effect on this, a negative effect of the intervention on infarcts, but actually it had a positive effect in men versus when compared to women. So there was an article, as I mentioned to you, Dr. Clayton published with Dr. Collins that called to action in 2014. Well, in 2020, there was an article on the 5-year progress to date with Dr. Clayton as a co-author or the lead author on this. And it was clearly indicated that we have made progress, that by putting these requirements into the NIH portfolio has had a positive effect, but that we still have a ways to go in improving adherence to this requirement and also improving the evidence as it relates to males and females, men and women. So we're going to see this.
It's incredibly important to consider sex as a basic biological variable in research because, well, half our population is of one sex or another sex. And we know from the science that there are many developmental aspects that influence biology according to sex. There are many life circumstances that influence biology according to sex, and there are changes in biology over a lifetime that influence our response to diseases and our response to life circumstances according to sex. So it's really important to consider sex. It's just a basic biological variable. You need to ask yourself, do you want to study only females? Is your question more appropriate only for males? You need to ask yourself if you're going to study females or include females. Do you want to study females in their reproductive life, or do you want to study them in reproductive senescence? And of course, rodents have something called reproductive senescence, which is actually not the same at all as human menopause. So if you're thinking about wanting to make your experiment relate to a human condition, you then have to ask yourself, is this the appropriate model? In order to think about the inclusion of females and males in animal work, we need to revisit some of our tried and true scientific assumptions. So one scientific assumption is that you don't report negative data. It's coming up over and over again in this meeting, this workshop, that we need to report negative data. We need to know when there isn't a difference as well as when there is a difference. And we need to understand the conditions under which the negative difference or the positive difference occurred. So a familiarity with the nuances of both the model you want to work with, what the animals themselves do, and also a familiarity with the nuances of hormones. I think a lot of people think that a hormone is a hormone is a hormone, but of course there's 17-beta estradiol, there's estrone, there's estriol. The question is, which are you going to study? Are you going to study them opposed with progesterones? Are you going to study them opposed with synthetic progestogens? What is going to happen? So it is complex, but there are many good review papers out there now because the field has been developing, I think, strategically with an eye toward this moment when the NIH, the Canadian Institutes of Health Research, and the European Union would begin to demand for the good of science that investigators begin to include males and females in their models. There isn't any condition in which this actually won't be important, but I think Alzheimer's disease is an incredibly important condition in this regard, and because Alzheimer's disease is a multifactorial disease unless you're studying early onset, and early onset only affects a very small proportion of people.
So I wanted to give you just a brief overview of how others are looking at this in the broader context and how important it is. So knowing that this is the policy, the practice, understanding the science that's driving this relatively new policy, we want to talk a little bit about where you put this in your application and where is it being captured, or where are reviewers looking for it, and where do you as future scientists need to make sure that you're including this. So we know it's primarily in the research strategy section of the NIH application. And the expectations for NIH are clearly outlined here, that you explain how the relevant biological variables such as sex, but it's not only sex, it includes age, weight, underlying health condition, are factored into research designs and analyses of studies of vertebrate animals and humans. Studies that only are relevant to one sex can be done, and you can provide a very strong justification on why, either based on the medical literature, based on the clinical presentation of disease, or your preliminary data that would support your focus on one sex. But you would have to provide that strong justification to be successful with your submission and your review of your application. So how do we get there? Knowing it has to be included, has to be relevant. The NIH has proposed the 4 C's of studying sex to strengthen science, and they outline them as consider, collect, characterize, and communicate. So in considering, this is how you're going to design your studies that take sex into account or explain why it's not there. When you're collecting your data, make sure that you have a mechanism for capturing sex and also tabulating it. In the analysis section, you can characterize the results based on the sex of the population or the samples that you had. And lastly, that you communicate by reporting either in oral presentations or in published works, the sex-based data as appropriate and relevant to your research question. So in considering it, how people get started is oftentimes you start with the very basics. You can do a literature search and look to see if clinically or in the basic sciences, are there sex gender variation. This can be simply done through PubMed or through the expanded samples for looking at literature. You can also look at your own data or data that you have access to see whether or not there is sex skewness in the disease incidence or prevalence that might suggest an underlying sex or gender-based influence, whether that be physiologically, pathologically, or otherwise. Certainly plan to include both sexes, males and females, in your test groups and looking at your randomization tables and presenting it as such. Report these differences and then conduct your pilot studies that would allow you to explore your hypotheses and determine whether or not your hypotheses are heading in the right direction. So here, it's also important, which I think that Dr. Clayton made this point, because some people say, well, why do I have to look for sex differences? What if there aren't differences? What if they're the same? The video clip also indicated this. Understanding that considering sex as a biological variable is not the same as looking for sex differences. It's just, and NIH is not going to require a certain design or a certain sample size or a certain method. They're going to expect the investigators to figure this out and to make sure that they have appropriately considered the potential effect of sex and planned accordingly. So looking at this, you want to make sure that you develop a data analysis plan that considers it. You want to report it, make sure that you've got a strong justification, and some of the reasons that people may give for supporting the justification. Certainly, if only one sex is scientifically appropriate, that one makes the most sense. For instance, if it's on ovarian cancer or prostate cancer, those would be perfect reasons to focus only on females if it's ovarian or males if it's prostate cancer. However, there may also be some other sex-specific conditions, but in cases people have made is that there are acutely scarce resources, and it could be that forever the study, it would be too expensive to do. In clinical studies, you cannot use cost as a mechanism, but here you could perhaps justify scarce resources as a means. Some of the challenges that have surfaced, and this began and was published in the peer review notes in 2016, and we still hear some of these challenges today, is that, what is convention and what is good science? You have to balance those in considering your progress and in your study design. It's also sometimes challenging for people to identify what is adequate incorporation of sex into their study designs. In the clinical realm, we've got a couple of decades of experience in it, but for the basic science, there's been some challenges. In some cases, people have brought forward that by increasing by including both sexes, you're increasing the physiological variability, and you might actually reduce the scientific rigor if you're not allowed to expand your sample size. So again, that's another balance. And of course, looking at whether or not if single-sex studies are more appropriate. And so therefore, you need to make that justification. Now, reproducibility also requires the reporting, and we talked about this in a little bit earlier. In preclinical studies, at the very least, the sex of the animals should be reported. And if only one sex of the animal was studied, this should be indicated, and they're saying it should be indicated in the title of the article, so that those consuming the medical literature will know at the get-go. In most cases, the sex of the origin of cells should be reported, but in some cell lines, perhaps that's not appropriate, but it's important to consider it and to be as transparent as possible. Clinical study designs now have pushed forward with not just saying you must include men and women, but that you should stratify randomly by sex whenever possible, so that the stratified analysis won't just be conducted post-hoc, but that you actually have strategically included it in your design. It's important that you, if you're looking for sex differences, that you have adequate statistical power to do so, because being underpowered might result in you finding nothing when actually not finding a difference when in fact there is one. Clinical study reporting is also very important in published research. And certainly if it doesn't, not just the title, but at least the abstract, should indicate whether a study involved only men or only women. If the study design also should indicate whether or not there was a identification of sex differences, and if there are, then those results should be presented. Oftentimes they're done in a supplement, but unless that is the primary reason for the paper. Researchers should be allowed to report inconclusive or descriptive sex-specific findings as raw data. Oftentimes this is done in an appendix, as I said, or as in a supplement, because oftentimes this can be used in meta-analysis to help us understand better about sex differences when an individual study may not have adequate power to detect differences that could be there. So in the grant applications, You would need to make sure that this is included in one of the sections of your application. And where this comes out in the required elements, for those of you who submit NIH research, you know that there are different sections of the application. There's the significance, the approach, the research strategy, these are the ones that are required for you to submit. You have elements of rigor, which are listed in the first column here, from the rigor of prior research, the scientific rigor, and then of course highlighted in yellow is consideration of sex and other relevant biological variables. Where you're going to see this in the section of the application, you would have to make the case in the research strategy. For the criterion score, which you see listed in the third column, where the reviewers will score each application based on their significance and their approach, the sex as a biological variable will be in the approach criterion because it's usually and most definitely going to be needed to include in the method section where you're describing your study design, your sample, your tissues, etc. This particular indication about sex as a biological variable does contribute to the overall impact score, and that is a specific question asked of reviewers when doing this. So if we were to look at this tree of review, let me just go over briefly with you how it would be addressed. So if the first question that reviewers would be looking at is, does the study involve vertebrate animals or humans? If the answer is no, then you don't have to consider sex as a biological variable, no brainer. If the question is yes, then it goes to whether or not the study intends to test for sex differences. If the answer is no, it doesn't mean that that stops there. You still have to ask the question, are both sexes included in the study? If yes, then the proposal needs to demonstrate the plans to report that data and disaggregated by sex. If the answer is yes, then this would be acknowledged as a strength and would be factored into the scoring. If it's supposed to demonstrate plans to report data disaggregated by sex and they don't do it, this would be acknowledged as a weakness and again would influence the score. If both sexes are not expected to be included in the study, then reviewers are going to look for the strong justification provided for the single sex study. If that is provided, this would be considered a strength. So even though it doesn't include both sexes, because they justified it, this would be considered a strength and would positively impact the score. And if it's not, then it would be considered a weakness and would also impact the score. For the situation where the study is not intended or is intended to test for sex differences, then the reviewers have to go to, again, the approach section, the analytics section, to see whether or not the design and the analysis adequately provided the rigor to test for sex differences. If that answer is yes, then it would be acknowledged as a strength and the scores would be influenced accordingly. And if not, if it was not designed or didn't have the analytic power to really detect these sex differences, this would be considered a weakness and would have the negative effect on the scoring. So in the language used in the review language for relevant biological variables, there are various things that one would be looking for in the review criterion strategy and in that approach. So have the investigators presented strategies to ensure a robust and unbiased approach? You can see other references to looking at sex differences in the sixth bullet here. Have the investigators presented adequate plans to address relevant biological variables such as sex? And then in the last bullet, looking at the questions on whether or not both sexes and genders, as well as inclusion of minorities and children, are part of the application. So NIH does put forth this mandate and this requirement, but yet it does it coupled with many resources to help us all understand how we can address this requirement and of course advance science in doing so. So there's several resources that if you just Google NIH policies and resources for sex as a biological variable, it will pull up things such as the ones listed here, not only the guidelines and the outlines, but also FAQs, as well as training modules that would be available. There are notices that have been distributed, workshops that have been held, analysis of public comments, and a number of research and training resources that can help you address this requirement and strengthen your applications. They also, the Office of Research on Women's Health also published an A to Z guide on sex and gender influences on health. This could be very interesting to you if you're trying to explore in that very first phase of considering sex as a biological variable, that you can get a quick look at where there may be some differences that would be important and relevant to your research. There are also many e-learning resources that are available. Here are some examples: Bench to Bedside, Sex as a Biological Variable, this primer that is in progress right now, as well as Introduction to the Scientific Basis of Sex and Gender-Related Differences. Again, these are coming soon, but there are many peer-reviewed publications already readily available on the websites to assist you. So my summary points are as follows. Certainly studying the effect of sex and gender will increase our understanding of human physiology and function. We know that there are sex bias factors that cause sexual bias in gene expressions through the genome. So although we know that the XXXY are important, we also know that the sex hormones have a very critical and important role to play in what happens. It is critical that researchers consider the impact of sex and gender on the interpretation of experimental results, and that the sex and gender research extends far beyond reproductive health, which is what we traditionally thought of when we thought about sex and gender research, but it has now become human health. So with that, I will conclude today's presentation and will be happy to entertain any questions. There's some questions there's something in the chat, but I don't know if these are questions. Okay, sounds okay.
Does anyone have questions for Dr. Krause-Wood? Unmute yourself or put them in the chat. Either way is just fine.
Okay, well, I'm not seeing any.
Questions? Okay. Well, Dr. Krause-Wood, thank you very, very much. We really appreciate that presentation. It was awesome. Thank you so much.
Well, thank you all. Appreciate the opportunity.
Thank you.
Don't know what the box was. Oh, that's your thing. Okay. All right. I will go ahead and leave you all. Thank you all so much for the time and attention.
Thank you again. All right, everyone. So I would like to ask you to log into VoxVote. You can see down at the bottom of your screen, it says live.voxvote.com. You can log in on a phone or on a computer or tablet. And once you log in, please put in the PIN that you see, which is 99802. This is the way we're going to have our little short questionnaire today so that you can vote on the few questions that we have to ask you. And it's all anonymous, so we ask you to please share your candid thoughts and opinions with us so that we can do our very best to make this course better and better every time we can offer it. So we'll go ahead and get started. Our first question is Thursday a good day for this class based on your schedule? And if not, what day would have been a better day for you based on your schedule? You can free text type in your response. And as soon as we get pretty good number of responses, we'll move on to the second question. Okay, so it looks like we're not getting any more responses. We'll go ahead and move on to the second question. Thank you, Tim. Was 3 p.m. a good time of day for this class based on your schedule? And if not, what would have been a better time of day for you? Again, you can free text type in. So again, was 3 p.m. a good time of day for this class for you? If not, what particular time of day would have been better for you? Okay, it looks like we're not getting any more responses, so we'll move on to the third question. Did you like having this class by Zoom, or would you prefer to have an in-person class? This is the first year that we've ever tried Zoom, obviously because of COVID reasons, but we're wondering if moving forward, if this is a good format for everyone, and maybe even after COVID, should we continue to offer the class by Zoom, or would you really prefer to be in the classroom with the lecturer each week? Tim, let's move on to the next question, please. Number 4. For those of you who would prefer an in-person class, which campus would be better for you? Is it easier for you to meet uptown, or would it have been easier for you to meet downtown? Again, if you would prefer the in-person class, which campus would be better for you, uptown or downtown? Thank you. Let's move on to question 5. What topics would you like to see added to the seminar series? So in a few minutes, we're going to ask you which ones you liked best or which ones you weren't as helpful. But for this question, I'd like to just focus on something that was not presented on. So if we were going to add a topic, what topic would you like to see added? Okay, I'm seeing research methods. That's interesting. Thank you. Mentorship. Thank you for that. Ethics. Let's move on to question 6, please. Which of the topics that were presented in the seminar series did you consider to be the most relevant? Thank you. Tim, let's move on to question 7, please. Which of the topics that were presented in the seminar series did you consider to be the least relevant, if any? We of course hope that they're all relevant, but we realize that they may not all be relevant to everybody who attends. So we just really would like to have your opinion about which of the topics weren't that relevant to you, to your course of study, to the type of research that you want to do, to your career. So if there's anything you'd like to share with us about that, we would certainly appreciate knowing that. So I'll just note as people are continuing to put their responses in here, if there's something that you think of after today's class that you would like to share with us, Brian and I welcome your comments. You can email either of us, you can call us, we'd be happy to chat with you. We really appreciate your feedback so that we can make this class the best that it can be. So please feel free to follow up with us. This questionnaire is not the only way that you can share your comments with us. All right, Tim, let's move on to our final question, number 8, which is very open-ended. Please give us any feedback you can about how our seminar series could be improved. We would appreciate any suggestions that you have for us. Okay, it looks like we're not receiving any more responses. So I would like to thank all of you very, very much for participating in the survey. We'll be reviewing the responses and doing our very best to implement changes that we can make to make the seminar series better next year. If you have any follow-up questions or issues, please feel free to call or email Brian or me, and we will do our best to assist you in any way we can. And I would just like to thank all of you for attending this year's seminar series. Thank you so much. And Brian, do you have any concluding comments?
It was a really great seminar series. So thank you everyone for your participation, and particularly the seminars where we had such great questions. So those I think really stood out, and I learned a lot as well. So I very much appreciate all of you. Thank you.
All right, thanks everyone. Bye.